Good morning, everyone. My name is Paul Thompson, and also on behalf of Andy Sakin, I'll be telling you a little bit about how the Phenotype Harmonization Consortium is facilitating data integration across many ADSP-funded efforts, and also making a lot of data ready for the AI projects. As you know, I, along with Tawfiq Raj and David Knowles, lead the AI Machine Learning Consortium for the ADSP, which is a set of 8 funded awards that are benefiting immensely from the data that's been generated by Tim, Michael, and Arthur in the Phenotype Harmonization Consortium. So just to recap, there's an extreme amount of interest in combining diffusion MRI, other types of imaging, cognitive data, vascular data, along with genomic and omics data to make predictions. And within AI, that's often making predictive models of patient outcomes or treatment response. But it may also be identifying endophenotypes or markers within the imaging data that is locked onto neuropath. And of course, to make these statistical models work, all of this data has to be available, curated, understandable, and made available to the many investigators that are taking on these challenges. So Andy and I have an advisory role in the PHC to help bridge some of the activities with the other ongoing work that's going on in AI/ML in particular. And you'll see a schematic of the way that the data is being handled and flowing into other efforts right here. So all of the aggregated landscape of biomarkers from imaging, fluids, neuropath is being harmonized within each domain. And so there are specialized approaches, some of which you've heard today, for harmonizing cognitive or vascular or PET or other types of data. But then once the derived measures have been created, these are integrated in the same computational space to discover novel markers of resistance or resilience to disease, models of disease progression, staging and subtyping, and then also subtypes that could be used for genomic analysis. If you could separate the component processes that are going on in aging and dementia, perhaps they're more refined substrates for genomic analysis. So I just wanted to say, there's many ways that the PHC is benefiting other activities that otherwise would find it very difficult to find the appropriate data in a curated form. There are ongoing synergies between the PHC and the so-called AI/ML consortium that's funded by the ADSP. And I'll give you a couple of examples in the next 5 minutes. So some are using multimodal imaging to predict neuropathology. Others are doing genetics on image-derived endophenotypes. Others are using the full force of AI to synthesize novel contrasts with imaging. Synthesizing PET scans is one of them. And the other is finally using genomic information to guide drug repositioning, which, as you know, is a major goal of ADSP. So here's one project where work from the Phenotype Harmonization Consortium has been immensely valuable. Dr. Dugu Tosun, working as part of the AI4AD initiative, is combining, obviously, the major demographic markers with structural MRI, amyloid, and tau PET, and other available data to train a machine learning method that will indicate whether or not a patient has particular types of neuropathology, TDP-43, Lewy bodies, or CAA. And she's really had tremendous success in using all of this data to provide pathologic information when it's not otherwise available, mainly to subgroup data for looking at stratification of drug trials, for example, and also to identify risk factors in the genome. You could screen people that fall into these categories for genomic markers that are associated with them. Now, just to be specific, she has been using ROSMAP, NAC, many of the other data sets that have neuropath as well as imaging. But you can imagine the whole roster of data freezers coming from PHC are going to be immensely helpful for this project, which usually is quite limited in terms of the available data. Other work that's being assisted by PHC is subtyping the imaging. So this work by Jinhao Wen and Christos Novatsikos is looking at all of the aggregated MR data from ADSP, using machine learning to define dimensions or subtypes, and then giving scores to individuals that then are amenable to GWAS. And they're finding individual genetic signatures for each of the subtypes, which normally are confounded or mixed together. And so you can imagine, again, all of the genomic data coming from ADSP and the associated curated imaging. You could do this not just for MR, but for PET, for FLAIR, for many other endophenotypes. And this is, again, a successful example of synergies between PHC and this AI effort. Another one that may not be familiar to everyone here is synthesizing new images. And so amyloid imaging is of great importance in AD research. You can also, to some degree, predict amyloid deposition from multimodal MRI. This is work by Yan Jin, in fact, our group, fusing all the multimodal data to synthesize PET. One of the things that is really needed is lots of paired MRI, FLAIR, PET data, and all of the associated clinical and cognitive biomarkers. And again, this project is benefiting immensely from PHC, giving new tranches of data that then can be used for training and testing and improving the synthesis of these novel contrasts. And then one final last project that's being facilitated by the PHC in the AI area work on genome-guided drug repurposing by Gunga Jun, Nathan and Dewal here, where they've identified particularly aggressive signatures in the genome that are associated with poorer outcomes in aging and AD. And they've done this by taking the AD GWAS and categorizing the loci by cell type, by pathway, and they identify, not just signatures associated with more aggressive progression, but modules that can then be linked via drug-gene interaction databases with treatments that particularly address some of the changes in these pathways. Now, one of the questions in this is that, are there really aggressive subphenotypes that can be stratified based on genomic analyses? And much of the data coming in from PHC is allowing us to address this for the first time. So this is an indication in the image of regions where there's ongoing atrophy, progressive atrophy, in people with this very aggressive subphenotype of AD polygenic risk, one that includes loci within a particular module that's expressed in microglia. So this partnership between Dr. Jun's group and also many of the investigators in the AI and ML projects are finding not just imaging correlates of genes, which is very, very important indeed, but demonstrating a biological correlate to the modules that are being identified using very advanced genomic analysis. So just as a word of gratitude, the first PHC data freeze was in October, primarily focused, as you know, on autopsy data, biomarker data, and cognition. These are already immensely valuable, as you know. The Neuropath project in particular is taking advantage of these. But we're also extremely grateful for Tim and Mike and Arthur's work, stratifying and phasing all of the future imaging data sets for input into the projects that I've talked about today. So you can look at the tremendous diversity here. There's YCAP, there's data sets with Neuropath, and there's huge data sets that have not been merged in the past. So just as a word of thanks, the point of this presentation was to point to ways in which the curated data is enhancing many other ongoing ADSP-funded activities. And we're certainly very grateful to the work of the PHC in enhancing and making it much easier to do the projects that we've discussed today. All right, thank you very much.